Hello students, welcome to EPG Pathshala. I am Dr. Sudhita Gautam Parehar from Hansraj College, University of Delhi. Today we are going to discuss about a module changing concepts of genes under paper molecular genetics. Learning objectives: gene, the fundamental unit of DNA, the concept of gene colinearity and gene organization, expression of genes, alternative splicing and its significance, pseudogenes and operon concept, central dogma and ribosomal genes. Introduction: gene within gene. DNA is the carrier of genetic information. Proteins are the building blocks of body made up of amino acids which are specified by the nucleotide sequence referred to as the gene. In 1902, Archibald Giroud suggested that genes code for the proteins. It was considered as the fundamental unit of DNA which codes for individual proteins. DNA is made up of nucleotides consisting of purines, pyrimidine bases associated with ribose sugar and phosphate molecule. The double helical structure of the DNA was revealed by Watson and Crick in 1953. The genetic information is transferred within the cell through a series of events known as replication, transcription, and translation. This concept of the flow of genetic information from DNA to proteins was referred to as the central dogma of molecular biology by Francis Crick. 3 years later, after deciphering the model of DNA structure, central dogma of molecular biology, which explains the genetic flow of information within the organisms in a biological system. This is a two-step process which tells about the passage of genetic information from RNA to DNA via transcription and finally to proteins via translation. This principle of central dogma explained that there is a one-way flow of genetic information from the DNA to proteins, which involves replication of DNA, passage of information from one DNA molecule to another, transcription, that is, formation of RNA using DNA as a template, and finally, expression of the RNA sequence into the proteins through the process of translation. RNA splicing is the process of removing introns and joining exons. Exon shuffling further complicates the concept of gene because the sequence of nucleotides which specifies each amino acid sequence of a protein exhibits alternative splicing. Ribosomes is a complex organelle formed by the association of several RNA molecules and proteins. It plays an integral role in the process of genetic information transfer from genotype to phenotype. Functional ribosome is made up of two subunits, one small subunit and second large subunit, each of which is coded by a complex unit of ribosomal RNAs and more than 50 proteins. Ribosomal genes are a brilliant example of multi-gene families. A multi-gene family consists of identical DNA segments usually clustered tandemly, and these sequences contain the entire ribosomal components. The identical DNA sequences are actually the genes coding for the ribosome's individual components. The entire set of genes are transcribed through a single transcription unit along the tandemly repeated hundreds and thousands times in a single or multiple clusters in the genome of the multicellular eukaryotes. This type of arrangement, that is, the repeated tandem molecules, is highly benefited as it helps many copies of the RNA transcription unit are capable of producing ribosomes at a very high rate of million molecules, which is required to meet the process of active protein synthesis. The primary transcript obtained is cleaved to yield the individual rRNA molecules. The individual molecules are then combined with the proteins and one kind of rRNA to form the ribosomal subunits. Pseudogenes, as the name suggests, are the false genes. That is, it refers to a gene which has lost its function and is not able to produce a functional gene anymore. Origination of pseudogenes is through the same mechanism as that of protein coding genes, which have undergone subsequent cumulative mutations that have led to the disruption in gene structure and hence their expression as functional proteins. As they belong to the non-coding part of the DNA, they are often designated as the junk DNA. With the recent advances in the molecular studies, it has been found that pseudogenes are present in various species ranging from plants, bacteria, nematode, insects, and even mammals. In humans itself, the number ranges from 10,000 to 20,000 pseudogenes. Operon concept was proposed in 1961 by Francois Jacob and Jacques Monod at the Pasteur Institute in Paris. Operon concept is the basic mechanism of controlling the gene expression in bacterial cells. The regulation takes place at the transcription level. In this case, the cells are themselves capable of adjusting the production level of certain enzymes involved in gene expression during transcription. For example, if we consider the availability of an amino acid, say tryptophan, the cell regulates its production by manipulating the enzyme's activities, which involve switching on and off of tryptophan production depending upon its level in the cell. In other words, the genes of the bacterial genome respond to the changes in the metabolic status of the cell. DNA, genes, proteins, central dogma, reverse transcription, pseudogenes, genetic expression in prokaryotes and eukaryotes. Concept of gene is so confusing. How it keeps changing. You have got a mail. It explains the changing concepts of genes to you. A mail? From DNA. Let's open it. The concept of gene colinearity. According to the concept of colinearity, the number of nucleotides in DNA should be proportional to the number of amino acids in the protein coded by that gene sequence. However, an ambiguity was observed in the DNA of the eukaryotes. It was found that the eukaryotic genes contain far more DNA than is required to encode the proteins. The extra amount of non-coding DNA is referred to as the junk DNA, which acts as a sink to absorb the mutations. Also, the mRNA produced by enzyme RNA polymerase using DNA template in the nucleus through the process of transcription was absent from the cytoplasm. The mRNA produced from transcription was much larger in nucleus than the mRNA present in cytoplasm. These observations of varying RNA sizes in the nucleus and cytoplasm led the researchers to suggest that nuclear RNAs underwent certain modifications before they were exported into the cytoplasm. Experimental evidence which proved the non-colinearity of the gene by hybridizing DNA and RNA. The concept of colinearity received a blow in 1970 when it was proposed that the 4 coding sequences in the gene of eukaryotic virus was not a continuous stretch but was interrupted by nucleotides that did not specify amino acids. The non-colinearity of eukaryotic gene was discovered by hybridizing DNA and mRNA. Electronic microscopic studies of the hybrids revealed that the size of DNA was much larger than that of the RNA. The results showed regions of DNA which were looping out from the DNA RNA pairs, and they proved clear evidences that the size of DNA was much larger than that of the RNA. These looping out segments of DNA were later on identified as introns. Alternative RNA splicing. Alternative splicing is the ability of free mRNA to be processed in various combinations to produce different variety of proteins through a single sequence of DNA. A single gene is capable to give rise to 2 or more mature RNA depending upon the joining of the exons. 2 types of alternate processing pathways have been identified. Alternative splicing. The pre-mRNA exons are shuffled in different combinations to yield multiple mRNA that are translated into different amino acid sequences and hence different proteins. By alternate splicing, exon 1, 2 and 3 are joined by removing introns to yield one mRNA. 2 exons, that is 1 and 3, are joined after removing the exon 2 and introns in between to give another mRNA molecule. Multiple 3' cleavage sites. The pre-mRNA has more than one potential sites for cleavage and polyadenylation. The pre-mRNA may or may not code for different proteins depending on the placement of the site before or after the termination codon. 1 and 2 3' cleavage sites. Cleavage may take place at the 3' end of site 1 or the 3' end of site 2. Two mRNA products of different lengths are produced after splicing, intron 1 and intron 2 respectively. Alternative splicing of calcitonin gene. A mammalian calcitonin gene exhibits both alternative splicing as well as multiple 3' site cleavage. The gene which codes for calcitonin has 6 exons and 5 introns. There are 2 possible cleavage sites present in the pre-mRNA, one after the 4th exon and second after the 6th exon. The entire gene is transcribed into the pre-mRNA. The product depends upon the site, that is, thyroid cells or the brain cells, where the RNA processing takes place. In the thyroid cells, the cleavage takes place after the 4th exon, producing a mature RNA consisting of exon 1, 2, 3 and 4, which is translated into the hormone calcitonin. However, if the RNA processing takes place in the brain cells, the cleavage occurs at site 2, that is, after the 6th exon. The mature RNA consists of exons 1, 2, 3, 5 and 6. On translation, this mature RNA produces a protein called calcitonin gene related peptide, a different form of calcitonin. Alternative RNA splicing in troponin T gene, which gives rise to 2 different muscle proteins. It is also observed in troponin T gene, which codes for 2 different muscle proteins. Researchers have discovered the central dogma stated that the genetic information flows in a single direction. However, with time, it was realized that the general flow of information includes the pathway of replication, transcription, and translation. But under special circumstances, it also includes information transfer from RNA to DNA in reverse transcription and transfer of information from RNA to RNA in RNA replication, which is observed in certain retroviruses and some transposable elements. Modifications of rRNAs into ribosomes in eukaryotes. A similar processing is also observed in eukaryotic rRNAs. The ribosomal genes 28S, 18S and 5.8S rRNA are clustered together, and this cluster of gene is tandemly repeated with untranscribed spacer DNA in between. The transcription units are present in large number. For example, in humans itself, the number is approximately 200. RNA polymerase 1 transcribes the entire unit within the nucleus. The promoter is present upstream of the 18S rRNA gene at -31 to +6, having an upstream control element of size 50 to 80 base pairs. Also, a Tata box binding protein or TBP is required for the initiation complex to transcribe the whole transcription unit. Precursor rRNAs undergo cleavage and trimming by RNAs to yield mature rRNA. Small nucleolar RNAs are involved in the cleavage, modification, and assembly of the processed rRNAs into mature ribosomes. Types of pseudogenes. Non-processed pseudogenes, which include the unitary pseudogenes and duplicated pseudogenes, which arise due to cumulative non-derelious mutations. Processed or the retro-pseudogene originate through retrotransposition. Therefore, they lack introns and promoter region, but often are flanked by polyadenylation tail. They often lack a proper regulatory environment, and errors in reverse transcription lead to the degeneration of processed copies of genes. Unitary pseudogenes are a result of mutations which affect the functionality of a gene. These mutations cause the loss of function in the gene without affecting the survival or fitness of the organism. These are non-deleterious mutations which are passed on to generations because the organism does not completely on this particular gene, which undergoes mutation to lose its function. The enzyme is that of the enzyme L-glucono-Y-lactone oxidase or GLO in primates. The GLO gene helps in ascorbic acid, that is vitamin C biosynthesis in mammals except guinea pigs. The GLO enzyme is not functional in primates due to pseudonization, and as a result the primates possess a GLO pseudogene. During evolution, there were mutations which corrupted the gene and there was no biosynthesis of vitamin C. However, primates, because of their omnivorous feeding habits, had a wide variety of foods to fulfill their requirement. This non-functionality of the gene could hamper their fitness. It has been observed that there are common mutations in the primate GLO pseudogene and humans. Thus, scientists claim that this suggests that all the primate species shared a common ancestor. The adaptation and migration with time to different habitats resulted in the divergence of different primate species. Duplicated pseudogenes comprise the largest class of pseudogenes. Molecular biologists suggest that in this case, the gene first underwent the process of duplication within the genome and later on mutations took place. The mutations in the copy of the functional gene were responsible for the loss of their functional properties and thus separated from the cell's functional machinery. There were adverse effects on the organism because all these changes took place in the duplicate part of the gene. Still, a fully functional copy of the gene was very much a part of the DNA to fulfill the requirements of the cellular machinery at the individual organism. The DNA of great apes and humans reveal many common duplicated pseudogenes which show identical mutations. Shared duplicated pseudogenes further strengthen the evolutionary relatedness of humans and the other primates. Processed pseudogene formation, a complex class of pseudogene which combines generation of DNA through the process of reverse transcription by a processed mRNA followed by its incorporation into the genome to give a processed pseudogene. Processed pseudogene is the DNA sequence obtained by reverse transcriptase. Any mutation in the inserted segment gives rise to processed or mutated pseudogene. Production of processed pseudogene 2 makes use of the cell's machinery to synthesize proteins. Once the newly generated DNA is incorporated into the genome, it provides large recombination of the introns and exons to alter the RNA message in several ways. The pseudogene produced in this way resembles the gene from which it has originated, but becomes non-functional due to the lack of proper information to initiate the production of the RNA message. The processed or retro pseudogenes lack the regulatory sequences and they are expressed only if they are able to align with some promoter region. A large variety of processed pseudogenes are shared by primates and human genome. Differences between processed and non-processed pseudogenes. Non-processed pseudogene are formed by direct mutations or duplications followed by mutations. Processed pseudogene are formed by reintegration of a DNA segment obtained through reverse transcriptases in the genome. Non-processed pseudogenes introns are present. Processed pseudogene, absence of introns and promoter sequences. Non-processed pseudogene, absence of a poly A tail. Processed pseudogene, presence of a poly A tail. Non-processed pseudogene, usually inserted adjacent to the original gene. Processed pseudogene, randomly inserted in the genome. Duplicated pseudogenes. DNA duplication generates copy of the original gene including introns, promoters and other transcriptional regulatory elements. Cumulative mutations result in the inactivation of the duplicated gene which becomes the pseudogene. Tryptophan operon. Tryptophan operon, also known as TRP operon, synthesizes amino acid tryptophan and consists of a multi-step pathway. The TRP operon consists of 5 structural genes which code for enzymes involved in the tryptophan biosynthesis, a TRP promoter and a TRP operator sequence. Each step in the pathway is catalyzed by a specific enzyme. All the enzymes are clustered together on a single bacterial chromosome. A single promoter is responsible for the activation of all 5 enzymes, which together constitute a transcription unit. On transcription, a single mRNA molecule is obtained which codes for all the 5 polypeptides constituting the individual enzymes which are required in the tryptophan biosynthesis pathway. The operon produces tryptophan when an RNA polymerase binds to the promoter and transcribes the gene of the operon. When the levels of tryptophan diminish, a TRP repressor protein encoded by a separate operon is produced which forms a dimer. The dimer form of the repressor is inactive or unable to bind the TRP operator, and thus the production of tryptophan continues as the structural genes of the TRP operon are transcribed. However, when tryptophan is present in the cell, there is no need for the expression of the structural genes and they are turned off. This switching off of the genes is achieved when the tryptophan binds to the TRP repressor to activate it. Activated repressor is capable to bind with the operator and stop transcription of the structural genes. This is referred to as a negative regulation, and the tryptophan molecule is known as co-repressor. If tryptophan is accumulating in the cell, more and more TRP repressor molecules are activated which can bind to the TRP operator and shut down the production of tryptophan biosynthetic enzymes. But if the levels are low, the transcription of the tryptophan genes is resumed. Tryptophan operon presents an example of how the changes in the cell's internal and external environment are able to regulate the gene expression. So students, let us summarize what we have learned in this module. DNA is made up of nucleotides consisting of purines or pyrimidine bases associated with a ribose sugar and phosphate molecule. In 1902, Archibald Garrod suggested that genes code for the proteins. It was considered as the fundamental unit of DNA. In 1958, Francis Crick said that genes and proteins are collinear. That is, there is a direct correspondence between the nucleotide sequence of DNA and the amino acid sequence of a protein. It was found that the eukaryotic cells contain far more DNA than is required to encode the proteins, and this junk DNA acts as a reservoir for the mutations. Eukaryotic organisms show the presence of polycistronic mRNA. Polycistronic mRNA refers to a single pre-mRNA which codes for a large number of proteins. Alternative splicing is the ability of a pre-mRNA to be processed in various combinations to produce different variety of proteins through a single sequence of DNA. It provides an opportunity of exon shuffling in the mRNA without changing the sequence of the exons and acts as an important source of protein variability in the vertebrates. Central dogma explained that there is a one-way flow of genetic information from DNA to proteins. The process of reverse transcription is observed in certain retroviruses and some transposable elements. This flow of genetic information is in the opposite direction from RNA to DNA than the usual flow, that is from DNA to RNA. Functional ribosome is made up of two subunits, a small subunit and a larger subunit, each of which is coded by a complex unit of ribosomal RNAs and more than 50 ribosomal proteins. Prokaryotic ribosome is a 70S molecule having a large and small subunit of 50 and 30S sedimentation coefficients respectively, whereas the eukaryotic ribosome monomer is 80S having 60S and 40S subunit respectively. Ribosomal genes are a brilliant example of multigene families. Pseudogenes is a gene which has lost its function and is not able to produce a functional gene anymore. In the evolutionary studies, they are referred to as molecular fossils obtained from once a functional gene. Operon concept, which is the basic mechanism of controlling the gene expression in bacterial cells, was proposed in 1961 by Francois Jacob and Jacques Monod at the Pasteur Institute in Paris. Tryptophan operon, also known as TRP operon, synthesizes amino acid tryptophan and consists of a multi-step pathway. Thank you.